Hello, good evening, both of you. We'll wait for 5 more minutes and then we'll initiate the session. Okay, so good evening, everyone. Let's start the session for today.
Excuse me, ma'am.
Yes,
Could you be a little more louder?
yeah, can you hear me now?
Better, ma'am.
Okay, yeah. Okay, so we'll Today we'll talk about the physiology of the respiration. Okay, so so let's start with the anatomy of the our respiratory system. Okay, so here you can see okay, so our respiratory system. So this is the image, and here if we want to divide it, we can divide our respiratory system in two zones. So first zone is the conducting zone. Okay, so this provides rigid and conductors for air to reach the sites of gas exchange. Okay, so the nasal cavity from where we take the air inside our respiratory system. Okay, so that also comes in a conducting zone. So these includes respiratory structure, for example nose and the nasal cavity, pharynx, trachea, bronchi. So these are the conducting zones. Okay, so our nose, the nostrils, then we have the nasal cavity, after that we have this pharynx and the trachea and the larynx. Okay, and then the trachea, it gets divided into our both of the lungs. Okay, so then comes our respiratory zone, where this which is the site of gas exchange. Okay, so it consists of bronchioles, alveolar ducts and alveoli. Okay, and respiratory muscles, so diaphragm and other muscles that promote the ventilation. Okay, so both of our lungs, the right lung and the left lung, and here where the trachea is being branched, so the left primary bronchus and the right primary bronchus. Okay, and this is our diaphragm, and uh, yeah, so this is the anatomy of our respiratory system. So basically we inhale the air which is outside. From outside we inhale using our conducting zone of our respiratory system, and the air which goes to our lungs, where the smallest units of alveoli are present, and they do the gaseous exchange when it comes with the contact of blood. Okay, and basically they oxygenate the blood, and now the oxygenated blood travels to our body via heart. So that cardiac system we have already discussed. Okay, so this is a section across our lungs. Okay, so here you can see the vertebra is there. Okay, and the anterior portion you have the sternum, in between both of the lungs you have the heart. Okay, and you have the pulmonary trunk and pulmonary veins. Okay, and these are the both of our lungs with the branches. Okay, so lungs, the soft spongy elastic weighing about 1 kg on surface hexagons. Okay, these are suspended with double walled pleural membrane. Okay, so the double walled pleural membrane, lobed left 2, right 3, and each lobe with a several bronchopulmonary segment bound by connective tissue, septa highly vascular and innervated by autonomous nervous system. Okay, yeah, and if we'll see from the trachea, it gets further divided and divided. Okay, so this is the trachea, then it again gets divided and again and again. So like this, the total branches is happening at 23 times. Okay, from trachea to the alveoli. Okay, so this is a schematic representation of the sequence of airway branches as a function of generation. uh, Z, and the bronchi, bronchioles, terminal bronchiole. Okay, so Z is the initial point, then we have the bronchi here. Okay, then we have the bronchioles. Okay, so bronchioles, and then we have the terminal bronchioles and the respiratory bronchioles, followed by the alveolar ducts. Here the alveolar ducts start, and at the end we have the alveoli sacs. Okay, and then how what are the cells that are situated, if we'll see. So the trachea, Okay, so from the throat towards the alveoli. So these are the cells that are present basically. So in the trachea and bronchi, so the epithelial tissue of the conducting airway serve as a channel for intake and exhalation of gases in the respiratory cycle, and are composed of mainly 5 types of cell. Okay, so we have the Clara cells, which are nothing but secretory cells. Okay, so in the trachea and bronchus, you can see, so there is one particular cell, this basal cells, which are uh, nothing but the epithelial cells, okay, which can be regenerated. Okay, and we have these green ciliated type of cells, okay, in the trachea, and we have the goblet cells. Okay, so the goblet cells are the cells that produces the mucus. Okay, so to keep our airways a little wet, okay, or a little lubricated, so these are the goblet cells which produces the mucus. And apart from that, we have Clara cells. These are responsible for detoxifying the harmful substances inhaled into the lungs. Okay, so these ciliated cells, they are a kind of special cells. Okay, so these ciliated cells are also present in our nose. Okay, in our nose also. So when we breathe the air, and if with the air some dust particle has come, so to avoid of it going to the our main respiratory system, uh, it sends it to the pharynx. There it goes to the esophagus and in the alimentary canal, because those dust particle can be taken care of the by the digestive system, but cannot be taken care by the our respiratory system. It can cause a very big problem. So that is why these trachea, these brushes have done in the nose also. They are present, these kind of ciliated cells, and they do the downward movement of that dust particle towards the pharynx, and it goes to the pharynx, those dust particle, and that's how it goes to the alimentary canal and does not go to the respiratory system. Okay, so the same cells which are present here in the trachea, the movement of the cilia here is not downward, it is rather upward. So in the nose, when the dust particle reaches, the downward movement of cilia takes place so that it goes to the pharynx. But when it reaches to the trachea, the ciliated cells do an upward movement so that the dust particle mixed with the mucus and reaches to the pharynx, and that's how the again from the pharynx it goes back to the alimentary canal. Okay, so that is the difference between both of those ciliated cells present in the nose and in the trachea. Okay, then we have the bronchioles. Okay, and then we have the terminal bronchioles. And yeah, so the functions of the respiratory passageway, trachea, bronchi, bronchioles. So one of the most important problems in all the respiratory passageway is to keep them open and allow easy passage of air to and from the alveoli. Okay, so to keep the trachea from collapsing, multiple cartilage rings extend about 5/6 of the way around the trachea. Okay, here you can see trachea and esophagus cross section. Okay, so in the walls of bronchi, less extensive curved cartilage plates also maintain a reasonable amount of rigidity, yet allow sufficient motion for the lungs to expand and contract. So these plates become progressively less extensive in the later generation of bronchi and are gone in the bronchioles, which usually have the diameters less than 1.5 millimeters. Okay, so for trachea and in the bronchia, initial bronchia, so there these cartilage rings are present which keep them um, uh, like uh, extensively flexed. Okay, so that they do not collapse with each other. However, when you go lower, so then the system is not there. So the walls of the conducting airways contain smooth muscles. Here you can see these are the smooth muscles which are present here. Okay, so in the normal patient, these are generally a relaxed smooth muscles. However, in asthmatic patients, these smooth muscles have become very tightened. Okay, so the walls of conducting airways contain smooth muscles, and these smooth muscles has both sympathetic and parasympathetic innervations. Okay, so when we were studying our muscle cells, okay, so there we have read about these kind of sympathetic and parasympathetic responses. Okay, so these smooth muscles have both sympathetic and parasympathetic innervation, so which have opposite effect on the airway diameter. Okay, so when the sympathetic adrenergic neurons, okay, so they activate this beta 2 receptor on the bronchial smooth muscles. Okay, and it leads to the relaxation and dilation of the airways. Okay, so in addition, and what is more important is, these beta 2 receptors are activated by circulating epinephrine released from the adrenal medulla, and by the beta 2 adrenergic agonists such as isoproterenol. That okay, so if the sympathetic nervous system gets activated, what does it do? It helps in the relaxation of these conducting airway. Okay, so sympathetic adrenergic neurons, okay, these activate the beta 2 receptor on the bronchial smooth muscle, and it leads to relaxation and the dilation of the airways. And in addition, what is more important, so the beta 2 receptor also gets activated by the epinephrine, which is released by the adrenal medulla. Okay, and second one is the parasympathetic receptor. Okay, so it is a cholinergic neurons which activate the muscarinic receptors, and it leads to the contraction and constriction of the airways. So sympathetic nervous system, if it gets activated, it causes the smooth muscle to relax, okay, for widening the airways. However, if the parasympathetic nervous system gets activated, it causes the airways to undergo constrictions. Okay, so in the asthma patient, you can see the airways are kind of blocked because of presence of mucus, a lot of mucus, so the airway is kind of blocked. So these patients are given um, the epinephrine like molecules. Okay, so that it acts, the epinephrine like molecules act on these smooth muscles, tightened smooth muscles, and try them to relax, to create some space for the airways for proper respiration in the asthma patients. Okay, so here you can see the respiratory bronchioles. Okay, so the trachea enters and the bronchi, and then we have the terminal bronchioles. Okay, then these terminal bronchioles further gets divided into the respiratory bronchioles. Okay, these are the respiratory bronchioles. Then we have the alveolar duct. Okay, these are the alveolar duct, individually they are branched. Okay, and then we these small uh, spheres are the alveoli. Okay, and these alveolar ducts open and forms the alveolar uh, alveoli, and all of the alveoli all together in a bunch, those are called the alveolar sac. Okay, so at the terminus you have the alveolar sacs, where you have the individual alveoli, and that is branched by the alveolar duct. Okay, so the alveoli location at which the gas exchange occurs. Okay, so these are the alveoli where the gas exchange actually occurs between the air and the blood. Okay, and 300 million or so provide huge surface area. Okay, so there are a lot of such alveoli are present, and because of that it gets so much more surface area. Okay, for the gaseous exchange to take place, and the anatomical dead space conducting pathways, passageways where no gas exchange takes place. Alveolar pores interconnect the adjacent alveoli. Okay, so you can see there are alveolar pores are also present. Okay, so these are so that they can get connected from the adjacent alveoli. So yeah, this is again the same thing, the branches. Okay, as along with that you can see the air is coming from the airway, from the trachea and the bronchioles. Okay, and also these alveoli, okay, the terminal alveoli, they are covered by the capillary beds. Okay, so here you can see this is a branch of pulmonary vein, and uh, here you have the bronchioles. Okay, and then this is the branch of pulmonary artery. Okay, and both are surrounding and making a capillary bed around the alveoli, and this is where the gaseous exchange takes place. Okay, here you can see the smooth muscles are present. Okay, these red ones are the capillaries. Okay, this is the alveolus, one alveolus, this is the alveoli sac, and along with that the yellow ones are the called as elastic fibers. Okay, so these elastic fibers maintains the elasticity of the these alveoli. Okay, so smooth muscle cells, these are out pocket alveoli. Alveoli are surrounded by fine elastic fibers. Externally, alveoli are cobwebbed by capillaries. Yeah, so this is a further more clear picture. Okay, so these cells you can see, these are the our alveolus. Okay, so here with the alveolus you can see, and these are the alveolar pores in the capillary. You have these red blood cells in capillary, and these are the alveoli, which is gas filled airspace. Okay, along with that if you will see, there is a section of type 1 cell of alveolar wall. So this is one a type 1 cell of alveolus. Okay, this one you can see, this one is a type 1 cell, which is nothing but squamous epithelial cells. Okay, so alveolar wall is a single layer of squamous epithelial cells with scattered cuboidal cells. Okay, okay, and we have this different color coded cells. Okay, so these are type 2 cells, and these are also called as surfactant secreting cells. Okay, so these type 2 cells secrete surfactants. So what are the surfactants? So as we have already told that these are the alveolar sacs. Okay, where the air comes from the external conducting airways. Okay, so the air passageways, so the air comes and filled with this the entire air sac or alveoli sac. Okay, and when the air comes, okay, and in the blood also, you can see the capillaries are lined. Okay, so in the blood also you have these water molecules, and also in the lining, some of the lining of the alveoli sacs is of mucus, so water is also present. Okay, so what happens when the water comes in the contact of air, there is a high tendency, okay, of increasing the surface tension. Because of the high surface tension, what happens, the air cavity, so the water droplets connects with each other. Okay, and that start filling the airspace with the water. Okay, because the air and water contact with each other tends to increase the surface tension. Okay, and fill the gaseous cavity with the water. Okay, so to avoid that surface tension, okay, to decrease the surface tension, these type 2 surfactant cells are there, which secretes the surfactants. So surfactants are nothing but a mixture of lipid molecules. Okay, so which are nothing but the different types of phospholipids, or these are basically detergents. Okay, so these surfactants, what they do, they reduce the surface tension on gas exposed surface. Okay, and alveoli bring air adjacent to capillaries, alveolar and capillary walls, and their fused basal lamina form respiratory membrane and air blood barrier. Okay, so here you can see this portion is the alveoli. Okay, and this portion is the capillary. Okay, this is an RBC, and here the gaseous exchange takes place. And this is the capillary endothelium, this one. And then we have this one, this particular is alveolar epithelium, this one. And then we have this border, which is fused basal laminae of the alveolar epithelium and the capillary endothelium. Okay, and this is the nucleus of type 1 squamous epithelium cell. Okay, so alveolar and capillary walls and their fused basal lamina form the respiratory membrane. Okay, this forms a respiratory membrane, and it is nothing but an air and blood barrier, and only allows the gaseous exchange to takes place. Okay, so from the alveolus, oxygen goes and bind to the hemoglobin present in the RBC, and the CO2 comes in the alveoli and being exhaled. Okay, again this is the same image. Okay, here you can see this is the alveoli space, this is the alveolus, this entire thing. Okay, and these are the capillaries where you can see RBCs are present. Okay, you have this type 1 alveolar cells, you have type 2 alveolar cells. Okay, and this is the fluid with the surfactants to decrease the surface tension. Okay, and yeah, one more thing is the macrophages. Okay, here also you can see these are the macrophages present. So what are does these macrophages do? So if so we have talked about the dust particles or undesired molecules which can hamper the airways. So either they are taken care by the nose or in the trachea with those ciliated cells which send those dust particle into the pharynx. So if the dust particle is escaped from both of the places, or whatever remaining has come to the alveolus, there these macrophages, it engulf those dust particle and they travel themselves to the pharynx and again goes to the alimentary canal. Okay, so that is why these macrophages are also present in the alveoli. Okay, so the alveoli contain the phagocytic cells, okay, called alveolar macrophages. Okay, and the alveolar macrophages keep the alveoli free of dust and debris because the alveoli have no cilia to perform this function. Okay, so macrophages fill with debris and migrate to the bronchioles where the beating cilia carry debris to the upper airways and the pharynx where it can be swallowed and expurgated. Yeah, okay. Okay, so pulmonary ventilation. So the ventilation mainly consists of the following. Okay, so inflow and outflow of the air. Okay, so basically the ventilation, what? The inflow and outflow of the air. Okay, so diffusion of oxygen, carbon dioxide between the alveoli and the blood. Okay, so when the blood enters, there is a diffusion of the O2 and CO2 between the air present in the alveoli and the blood which is traveling in the capillaries. Okay, so transport of gases in the blood and the body fluids and the regulation of ventilation. Okay, so these our lungs are the, if you'll see, okay, so um, so these are the trachea. Okay, and then after the trachea branches, our lungs are there. Okay, so there is no support, or we can just say that the lungs are our lungs are hanging in a position. Okay, only joined to the body via the trachea. Okay, so so the lung is an elastic structure. Okay, and it collapses like a balloon. Okay, so these are um, basically hollow. Okay, and it has the property that it can collapse, okay, like a balloon, and expels all its air through the trachea whenever there is no force to keep it inflated. Okay, and also there are no attachments between the lung and the walls of the chest. Okay, so our lungs are present in situated in the rib cage. Okay, but there is no attachment. Okay, so except where it is suspended at its hilum from the mediastinum. Instead, the lung floats. Okay, it basically floats in the thoracic cavity surrounded by a thin layer of pleural fluid that lubricates the movement of the lungs within the cavity. So if the chest wall is opened, lungs will collapse. Okay, so how does the lungs float and does not collapse? So what happens? So we talked about the pleural cavity. Okay, in the thoracic cavity surrounded by thin layer of pleural fluid. Okay, so it lubricates the lungs. So basically in the pleural fluid, in the pleural cavity, you have a set of negative pressure. Okay, a negative pressure which is making the heart inflated. Okay, because of the negative pressure, it is like uh, stretching or making it inflated, not allowing it to collapse. So because of the negative pressure, the lung is inflated. Okay, and we have the respiratory muscles. Okay, so we have the diaphragm. So diaphragm is basically it is a dome shaped when it is relaxed. Okay, it is a dome shaped structure. You have the ribs. So ventricular flow is driven by pressure differences between the alveoli and the atmosphere. Okay, so the oxygen flows from higher partial pressure to lower partial pressure. Okay, so active contraction of skeletal muscles. Okay, so the rib cage is nothing but the skeletal muscle. Okay, generates this pressure during inspiration. Expiration results from the passive elastic recoil of the lungs. So the diaphragm is the main muscle of inspiration. Okay, so when you inspire, the diaphragm is the main muscle, so the most important muscle for resting breathing. So diaphragm is innervated by the phrenic nerve, and which originates from the third, fourth and fifth cervical spinal cord. Okay, and the contraction of diaphragm flattens the floor of the thoracic cavity. So what happens when the diaphragm, in the normal state it is in a dome shape. Okay, but when you do the inspiration, the diaphragm flattens. Okay, when the contraction diaphragm takes place, so in the relaxed state it is in a dome shape, but when it is contracted, it becomes flattened. Okay, it becomes flattened, and because when it becomes flattened, the thoracic volume increases. Okay, in the thoracic cavity the volume increases so that the outside air can be inhaled. Okay, and the rib cage also goes a little forward. Okay, rib cage goes forward. So here you can see during the inspiration the diaphragm contracts and flattens, and the external intercostal muscle raise the ribs to expand the thoracic volume, and during expiration the diaphragm again relaxes to its dome shape, and external intercostal muscle pull the rib down to decrease the thoracic volume. Okay, the external intercostal muscles also contribute to inspiration. Intercostal muscles are innervated by intercostal nerves from the thoracic segment of the spinal cord. Okay, so this the phrenic nerve. Okay, so here you can see the cervical vertebrae. Okay, so we have how many cervical vertebrae? 7. Okay, so we have 7 cervical vertebrae, and out of those, this the C3, C4 and C5. Okay, these the nerves which are coming to these vertebrae, okay, those spinal nerves, okay, they combine and originates a phrenic nerve. Okay, and in this phrenic nerve it innervates the diaphragm. Okay, so yeah, so the basic function of the respiratory system is do the inhale exhale. So, but how this process is being regulated, because these are the not involuntary muscles, these are regulated by the brain. Okay, so the brain sends the signal to the spinal nerves, which are these C3, C4, C5, which is ultimately the phrenic nerve. So the brain sends the signal to the phrenic nerve, which is innervating the diaphragm, and the diaphragm is getting signal of getting contracted. Okay, so because of that, the rhythmic contraction and relaxation of the diaphragm allow the inhalation and the exhalation process of our respiratory system. Okay, so the transaction of the spinal cord above the third cervical segment is fatal. Okay, but the transaction below the fifth cervical segment is not. Okay, so if you take a knife and make a cut. Okay, so if you make a cut below the C5 segment of the cervical segment or the spinal cord, okay, or the cervical vertebrae, it is not fatal. However, if it is above that, it is fatal because it will ultimately damage the phrenic nerve, okay. But the transection below the fifth cervical segment is not, because it leaves the phrenic nerve that innervate the diaphragm intact, okay. So any transection in the above range of this cervical vertebra or in this spinal nerve above the C5 is fatal. However, below the C5 it's not fatal, okay. Yeah, so as I was talking about the pressure, okay, so that the negative pressure that keeps the lung inflated, okay. So here you can see, so this is the interpleural pressure, which is in a negative pressure, okay. So here you can see, this is the lung, this is the pleural space, and this is the chest wall or the rib, okay. Okay, so if you see the pressure, so the pressure level that we are measuring here, so yeah, so here we are measuring the pressure in centimeter water, okay, not the mmHg pressure, okay. And this is the volume change, okay, the thoracic volume change, okay. So how much air is coming, okay. So here you can see, so initially if we'll see the pressure, the volume 0, if we'll consider the volume 0, okay, almost 0, then you undergo inspiration, okay, the inspiration of air is happening, then you can see there is an increase in the volume, okay, in the volume, the lung volume or the thoracic volume you can say, okay, there is an increase. So from 0, how much it is going? It is going 0.5 liters, okay, that means 500 ml, okay. So when you inspire, when you inhale, then you if the volume of the cavity increases and the 500 ml of volume comes to the lung, okay, 500 ml of air it is coming, okay. And in the again in the expiration, the lung volume again decreases, so that 500 ml that you have taken is now released, okay. So that is inspiration and expiration cycle. So in every inspiration you take 500 ml of air, and then in the expiration you releases that 500 ml. So in that case what is happening, so you have two different pressure. One is the alveolar pressure, here in the alveoli, the alveolar pressure, and then you have the pleural pressure, okay, the fluid cavity, uh, the cavity which is filled with the pleural fluid, okay. So you have the pleural pressure and you have the alveolar pressure, okay. So um, uh, this alveolar pressure also initially it's near 0, okay. And then as the inspiration is happening, so that also has to create a negative pressure so that the air can travel from high pressure to low pressure, right. So when you do the inspiration, there is a from 0 to -1 the pressure is created, okay, in the alveoli, okay. So in the alveoli you have the -1 centimeter water pressure, yeah. So in the alveoli you create a negative pressure which is -1 centimeter water, that negative pressure, and that pressure allows to inhale 500 ml of air, okay. So that much air is or the that much volume is being changed, so 500 ml air can be inhaled with a -1 centimeter water negative pressure, okay. So that's how the inhalation of air takes place. And when you have to exhale or expire that air, then again the pressure increases +1, okay. So the alveolar pressure increases and the volume is again brought back to 0, okay. And at the same time, so you have this pleural pressure. Okay, so as I have already told, to avoid the lungs to collapse, okay, you already have a negative pressure in the pleural cavity, so that it does not collapse, the lungs does not collapse. So you already have the some -5 water pressure in the pleural cavity. Okay, so when you do the inspiration, uh, the alveolar pressure decreases, and also this pleural pressure decreases to provide more space, or to um, inflate the lung, to increase the thoracic volume. Okay, so that the 500 ml of air can come in. Okay, so again it goes to the negative, and again goes back to the initial state when the expiration is happening. Okay, and the difference between these alveolar pressure and pleural pressure is nothing but the transpulmonary pressure. So this is what is happening, okay, when you do the inspiration and expiration. So yeah, so then one more thing comes is the compliance of the lungs. Okay, so I've shown you the alveoli, okay, along with the capillaries, it's are also wrapped with the elastic fibers. Okay, so that provides them the elasticity, okay, how much the alveoli can expand. Okay, so the extent to which the lungs will expand for each unit increase in the transpulmonary pressure, okay, is called the lung compliance. Okay, so the total compliance of both lungs together in normal adult human being averages about 200 ml of air per centimeter of water transpulmonary pressure. So this is every time the transpulmonary pressure increases 1 centimeter of water, the lung volume after 10 to 20 seconds will expand 200 milliliters. Okay, this is the compliance of lung. Okay, so that also depends on the elasticity. Okay, so if uh, suppose your lungs are not that elastic, okay, and due to that the compliance of your lung is changed. Okay, so compliance can be considered the opposite of stiffness. Okay, so the a low lung compliance would mean that the lung would need a greater than average change in the interpleural pressure to change the volume of the lungs. Okay, and a high lung compliance would indicate that the little pressure difference in the interpleural pressure is needed to change the volume of the lungs. So more energy is required to breathe normally in a person with low lung uh, compliance. So person with low lung compliance due to disease therefore tend to take shallow breaths and breathe more frequently. Okay, yeah, so 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 what happens in the disease condition, what happens, so the lung, they become fibrous, okay, because of the what you say, the increased mucus and everything in the asthmatic condition. Okay, so the with increased mucus the lungs, it becomes fibrous. Okay, and it loses its elasticity, and because of low elasticity the compliance also becomes low. So if there is a low compliance, means more intrapleural pressure to take to change the lung volume. Okay, low lung compliance would mean that the lung would need a greater than average change in the interpleural pressure to change the volume of the lung. Okay, so person with low lung compliance due to disease therefore tend to take shallow breaths and breathe more frequently. Okay, so the effect of pulmonary fibrosis on lung architecture. Okay, this is the normal lung, and this is the pulmonary fibrosis. Okay, if and this is the lung architecture. Yeah. So this is uh, just to say the pulmonary volumes. Okay, so the pulmonary volumes and recording capacities, recording changes in pulmonary volumes, spirometry. Okay, so it is a simple method for studying the pulmonary ventilation. okay, is to record the volume movement of air into and out of the lungs. Okay, so this is a process called spirometry, and it consists of a drum inverted over the chamber of water, so with the drum counterbalanced by a weight, and in the drum is a breathing gas, so usually air or oxygen. A tube connects the mouth with the gas chamber. When the one breathes in and out of the chamber, the drum rises and falls, uh, and an appropriate recording is made on a moving sheet of the paper. So this is the figure. Okay, so this is a drum which is a counter balance with this weight. Okay, this has no weight of its own. It is a floating drum. Okay, this is the water where the oxygen chamber is present. Okay, so this is a mouthpiece where you can blow the air in this spirometer. Okay, and this will record. This is a recording drum. This will record the volume changes. Okay. Okay, for ease in describing the events of pulmonary ventilation, the air in the lungs have been divided into this diagram into 4 volumes and 4 capacities. Okay. Okay, so we have read, like, with each inspiration, the lung volume increases to 500 ml. Okay, so but at the normal condition also, which is this line, okay, here you can see you already have 2300 ml of lung volume already present. Okay, 2300 ml lung volume already present. Then you undergo respiration, inspiration. Okay, the normal inspiration, and then you inhale more. Okay, a lot of volume, as much as you can inhale, you do the inhalation. Okay, and that's how so this is in the normal condition. From 2200 to 2800 lung volume increases. Okay, then when you inhale a lot of air then the lung volume can goes up to maximum up to the 5800 5700 ml. Okay, and then you decrease, and these are the, again you decrease or exhale as much air as you can. So then also the maximum you can exhale, still there is some 1200 or 1100 ml lung volume remains. Okay, so these are the different capacities, okay, of the lung. Okay, so maximum inhalation, that is called as inspiratory reserve volume. Okay, inspiratory capacity, and this is the vital capacity, maximum inhalation and maximum exhalation, and this is the total lung capacity. So maximum goes up to 5800 700. So the vital capacity can be used to help differentiate causes of lung disease in restrictive lung disease, and the vital capacity is decreased. Okay, so this is the vital capacity. Okay, the maximum and the minimum. Okay, so this is used to help to differentiate the cause of the lung disease. Okay, and this diagram shows the volume of alveolar air replaced by new atmospheric air with each breath is only 1/7 of the total. Okay, so that multiple breaths are required to exchange most of the alveolar air. Okay, so this figure shows the slow rate of renewal of the alveolar air. So this is the alveoli, these are the air molecules. Okay, so in the first alveolus of the figure, the excess amount of gases is present in the alveoli, but note that even at the 16th breath, the excess gas still has not been completely removed from the alveoli. So you need successive rounds of breath to completely remove the already present air and fill with the new one. Okay, so the average male functional residue capacity of the lung, so the volume of air remaining in the lung at the end of the normal expiration, measures about 2300 ml. Okay, yet only 350 ml of new air is brought into the alveoli with each normal inspiration. Okay, and the same amount of old alveolar air is expired. Okay, therefore the volume of alveolar air replaced by the new atmospheric air with each breath is only 1/7 of the total. So the multiple breaths are required to exchange most of the alveolar air. Okay, so the slow replacement of alveolar air is particularly important in preventing sudden changes in gases concentration in the blood. Okay, this makes the respiratory control mechanism much more stable than it would be otherwise, and it helps prevent excessive increases and decreases in tissue oxygenation and tissue carbon dioxide concentration, tissue pH, and other factors. Okay, so this is the mechanism of gas transport in the final branches of the mammalian lungs during inhalation. So here you can see two types of arrows are there. Okay, so one the straight arrows which shows the pathway of the air that is being traveled. Okay, and once it is coming in the alveoli, what is happening, the this motion less air is present. Okay, so this air is not making any motion, but only diffusion of the air is taking place in the alveoli. There is no motion is taking place. Okay, yeah, so surfactant we have already talked about. Okay, so this is the layer of surfactant that is being released by the type 2 cells. Okay, the type 2 alveolar cells. So this is type 1 alveolar cells present. This is type 2 alveolar cell which is releasing the surfactant to reduce the surface tension. So surfactant is a surface active agent in water, which means that it greatly reduces the surface tension of water and is secreted by the special surfactant secreting epithelial cells called the type 2 alveolar epithelial cells, which constitute about 10% of the surface area of the alveoli. Okay, yeah, so the surfactant is important at birth. So the fetus makes a respiratory movement in the utero, but the lungs remain collapsed until the birth. Okay, so after the birth, infant makes several strong inspiratory movements and the lung expand. So surfactant keeps them from collapsing. Okay, so surfactant deficiency is important cause of infant inspiratory distress syndrome. So the condition precipitates if the surfactants are not adequate. Okay, and exogenous surfactant replacement therapy is effective in reducing RDS, which is respiratory distress syndrome, mortality and morbidity in the preterm infants. Okay, here you can see this is the muscle, this is the airway wall, and this is the thickened airway wall muscle because of the presence of the mucus. Okay, asthma is a common chronic inflammatory disease of the airways characterized by variable and recurring symptoms. Okay, reversible airflow obstructions, bronchospasms. Common symptoms includes wheezing, coughing, and all. Okay, yeah. This is epinephrine molecule, and this is salbutamol. Okay, okay, so you can see structure wise they are almost similar. So this is epinephrine, so in place of epinephrine salbutamol can be given. Okay, so which is for the treatment of acute symptoms, and usually with an inhaled short acting beta 2 agonist. Okay, so, and oral corticosteroids. Symptoms can be prevented by avoiding triggers such as allergies and irritants. Okay, so salbutamol is a short-acting beta-2 adrenergic receptor agonist. Okay, used for the relief of bronchospasm in conditions such as asthma and chronic obstructive pulmonary disease (COPD). Okay, so we have talked about the sympathetic and parasympathetic nervous system. So in case of sympathetic nervous system, for the relaxation of the bronchioles. Okay, so of the smooth muscles. Okay, which in the treatment condition becomes very tightened. Okay, so you can give the so epinephrine was there to help to relax those muscles. Okay, for the relaxation and making the airway space broader. Okay, so in place of epinephrine, salbutamol can be given, which acts as a beta 2 adrenergic receptor agonist. Okay, yeah, yeah. So after this, maybe we'll talk about in the next session. Okay, so based on that, we have the questions ready, which we can see here. Okay, so here is the lung and the branches of the trachea that goes to the lung. Okay, so what is the second one? So these are already shown. These are the respiratory bronchioles. These are the alveoli. So what is the second one?
Alveolar duct.
No. So from the trachea it branches to the bronchus, right? The bronchus and the bronchioles. Okay, then the alveoli and alveolar duct start. Okay, so this is the terminal bronchiole. Okay, this particular portion is the terminal bronchiole. Okay, this one. And after the terminal bronchiole, then we have the respiratory bronchioles. Okay, the terminal bronchioles, then we have the respiratory bronchioles, then the first one and also the third one, they are showing the alveolar ducts. Okay, because they possess the alveoli. So these are the alveolar ducts, first and third. Okay, first and third both are alveolar ducts. And what is the, and the second one is the terminal bronchi. Okay, and this is the alveoli, and all the alveoli together, these are called as alveolar sac. Okay, yeah, next we have this cross section of the alveoli. Okay, so this one shows the fluid with the surfactant. Okay, and then what is the third one, this one. So this sac or this air filled cavity is called what?
Alveolum.
It is alveolus. Okay, a single one, that is why alveolus. If there are multiple, they are alveoli. Okay, so this entire cavity is alveolus, where the gaseous exchange will take place. Okay, so because here in the alveoli we have the air that will come from the air passageway. Okay, and where the gaseous exchange will happen. So gaseous exchange will happen with the RBC. Okay, so this particular section which is carrying the RBC, these are what? These tubules are what?
Capillaries.
Yeah, these are the capillaries. Okay, we have also the white blood cells, and then these are the second one. What are these?
Alveolar cell type 2.
Yeah, these are the type 2 alveolar cells, and what does they do? What is their function? They release the surfactants, right? So these are the type 2 alveolar cells which releases or secretes the uh, surfactants. Okay, so surfactants includes the lipid dominant molecules or detergent, you can say. Okay, so those surfactants, what they do, they help in reducing the surface tension, so that when the air and water comes in the contact with each other, it should not make a bond with the water molecules should not make a bond with each other and fill the cavity with the water. So to avoid that, the to decrease the surface tension, these surfactants are being released from the type 2 alveolar cells. And then we have the squamous epithelial cells in this particular alveolus, which are called type 1 alveolar cells. So, okay, so this one is type 1 alveolar cells, this is the type 2 alveolar cells, this is an alveolus, and this is a capillary. Okay, okay. So what is this?
Diaphragm.
No, this is a diaphragm. Okay, the here. So these are the lungs, and below the
Lungs.
lung we have the dome shaped diaphragm. Okay, okay. So diaphragm movement is regulated by brain. Okay, by which particular nerve? So the
Phrenic nerve.
yeah, phrenic nerve.
Phrenic nerve.
Yes, phrenic nerve. Okay, so these are the cervical vertebrae, C3, C4, C5. Okay, those cervical vertebrae which where the spinal nerve goes, and these three spinal nerves all combined together, they make the phrenic nerve. Okay, so here at the right side we have the right phrenic nerve, left side we have the left phrenic nerve. Okay, and this phrenic nerves only go and innervate with the diaphragm and control the relaxation and the contraction of the diaphragm for the inspiration and the expiration. Okay, so yeah, the first one here is the, so this is the right phrenic nerve and this one is the left phrenic nerve. Okay, here we have our heart. Okay, this one is lung, and the fourth one is,
Diaphragm.
yeah, fourth one is diaphragm.
Diaphragm.
Yeah, good. Okay, so what are these two molecules? The first one is,
Salbutamol.
no, first one is epinephrine. Okay, first one is epinephrine, second one is salbutamol. Okay, so what is the function of these two in aspect of respiratory system? Okay, so we have talked about the sympathetic and parasympathetic nervous system. Okay, so the bronchioles, there are smooth muscles are there, okay, on the bronchioles, which are innervated by the sympathetic and parasympathetic nerves. Okay, so there we have the relaxed smooth muscle. In a normal person you have the relaxed smooth muscles. Okay, but when there is a disease condition you have the tightened, very tightened smooth muscles. So what happens, so in different kind of situation you have different responses. So when your sympathetic nervous systems gets activated, okay, so it acts on the beta 2 adrenergic receptor. Okay, it releases the epinephrine. Okay, so what does epinephrine do? It go and bind to the beta 2 adrenergic receptor present on the smooth muscles, okay, which are situated on the bronchioles. Okay, okay,
Ma'am, beta 2 blockers are.
so beta 2 blockers are for heart. Okay, those are different. Okay, but the receptor I'm saying is the same. Okay, so the epinephrine receptor, okay, which is present on the um, bronchioles or in the respiratory system, that is beta 2 adrenergic receptor. So epinephrine goes and bind there, and what function does it perform? It causes the relaxation of the smooth muscle. Okay, so relaxation means broadening the airways. Okay, so that the more air can come. Okay, so broadening the airways. So what happens in disease condition, what you have, your airways are filled with mucus. Okay, so in those condition also you need kind of relaxation of the smooth muscles. Okay, so in that scenario you use an epinephrine like molecule, which is nothing but the salbutamol. Okay, this salbutamol, which is a beta receptor agonist. Okay, so which causes the relaxation. Okay, so it work as epinephrine, causes the relaxation of the smooth muscle, broaden the airways for easier breath intake. Okay, because the air passageway is relaxed and broadened. Okay, is it clear? Okay, okay, so this is all for today. We'll meet again in the next session. If you have any doubt you can ask me.
No doubts, ma'am. Thank you for your.
Okay, okay, thank you for attending the class. We'll see you again next week. I'll conclude
Okay,
the
ma'am.
session.
Thank you, ma'am.